Good afternoon, everyone. Welcome to our MHRC seminar, the first of the new year. I'd like to welcome today Dr. Vahab Soleimani from McGill University. Give you a little background on Dr. Soleimani. He got his Bachelor of Science degree in biotechnology from the University of Ottawa, and he did his PhD in molecular biology, studied transposable elements in the evolution of the genome. Then he joined Michael Rudnicki as a postdoc in 2007. He worked on transcriptional control of myogenesis. And then in 2014, he joined the Lady Davis Institute in Montreal and McGill University's Department of Genetics as an assistant professor, and now he's an associate professor at McGill in Department of Human Genetics. So it's great to hear this topic. We're going to hear about functional interaction between skeletal muscle stem cells and their niche environment. I'm anxious to hear more about the niche in development and diseases. So Dr. Soleimani, if you wouldn't mind, please turn on your video, and I will stop talking and welcome to our series this afternoon. Please share your screen with us and take it away.
Thank you, David. Thank you for the kind introduction and also for the invitation to present our latest work to your wonderful group in Toronto. I just want to make sure that my voice is coming through and then you see my slides. Is that the case?
Yes, we're good on the slides and your voice is good. Maybe talk a little louder. That would be great.
Great.
Yep.
So a key focus in our lab is to try to understand how muscle stem cells behave within their niche environment. And today, the entire focus of today's talk, which is going to be two parts, will be on trying to decode those molecular interactions that takes place in terms of communications between satellite cells and their neighboring non-myogenic cells and niche factors. So I know this is a very specialized group and I don't need it to make a lengthy introduction about what skeletal muscle is. So I only have this slide basically as an introduction. This is just for students in case the students are in the audience. Skeletal muscle evolved for the production of force and contraction. It's by far the largest organ in the human body. 40% of an average, the weight of an average individual is composed of skeletal muscle. This elaborate biophysical structure is actually formed by fusion of myogenic progenitors during embryonic myogenesis. Myogenesis has three distinct phases: embryonic, fetal, and adult myogenesis. And this myofibers then are basically the result of the fusions of numerous of these myogenic precursor cells or progenitor cells that themselves are composed of these repeating units called sarcomeres. These are the contractile unit of skeletal muscles, which themselves are composed of actin and myosin's motor proteins. By postnatal day 21, these myogenic precursor cells will exit cell cycle and then it'll acquire a satellite position around the muscle fibers. And these cells will be responsible for their wear and tear and the repair of the muscles throughout adult life. So at this stage, these cells are almost all of them going back to the quiescence, and then the growth of muscles from P21 into the adulthood mostly occurs through hypertrophy. So although these satellite cells or myogenic progenitor cells are absolutely critical, or the base building blocks of the skeletal muscles, they do so in cooperations with a lot of non-myogenic cells that are present within the muscle tissue, and that's going to be entirely the focus of my talk today. So this is a simple, relatively simple schematic cartoon drawn by one of my PhD students in the lab, illustrating what the muscle stem cells and its relation to the non-myogenic cells looks like. So you have the fiber right in the middle of this, sort of like an Easter egg, I call it. Satellite cells closely juxtaposed with muscle fibers. You've got the ECM covering the satellite cells. So this is what we define as anatomical niche of the muscle stem cells, very closely juxtaposed with muscle fiber. Within this environment, you've got all kinds of different non-myogenic cell types. You've got T cells, you've got fibroblasts, macrophages, and you name it. So all these cells within the past 20 years, we know that they've been shown that they actually directly or indirectly contribute to the function of muscle stem cells, whether it's through its secreted factors, or for example, in the case of macrophages, in terms of contributing to the ECM components like fibroblasts, endothelial cells, for example, providing notch ligands to keep satellite cells in a quiescent states. Muscle stem cells also get its cue from the systemic factors, from the factors that are present in the bloodstream. And this fiber itself, Aralab and then others have also shown that it's a critical signaling component of the muscle stem cell niche environment. So if you look at the possible pairwise and combinatorial interactions between muscle stem cells and any of these individual different cell types, you just appreciate the enormous complexity that might be taking place in terms of the signaling pathways that actually takes place between these cell types. So this is what we call it, the cellular complexity of the niche environment, but that's not the only component that contributes to the complexity of the communication between satellite cells and niche environment. The other component of the niche is the dynamic nature of the niche. The niche is not static, it changes quite dramatically during the course of diseases. For example, I'm sure most of you or all of you have seen cross sections or the muscle biopsies from patients with muscular dystrophy or the dystrophic mouse models, and how enormously the niche changes or the muscle composition changes during the course of the disease progression. But the muscle also changes in the absence of diseases. For example, a simple process of aging leads to dramatic changes in the muscle stem cell niche. So what I want to talk about today are two. One, I will talk about how the niche environment, the muscle niche environment contributes to the altered transcriptome and epigenome of muscle stem cells. And the second part, I will talk about how stem cells communicate with the fibers. So basically how the muscle fiber acts as a ligand and satellite cells as receptor, and what is the biological implication of these interactions between the myofiber and satellite cell is. So the first part, we are trying to understand the effect of aging muscle niche on muscle stem cell numbers, on the population dynamic of muscle stem cells, and also on the alterations in transcriptome and epigenome of satellite cells or muscle stem cells. Now, on the right, what I'm showing you here is simple fax plot from a 1-month-old mouse and a 24-month-old. So you can see these green cells, there's actually quite a dramatic reduction in the number of muscle stem cells as the mice ages. So we know that we've known this for many, many years. We know that it's not only the number of muscle stem cells that are actually reduced during aging, but also their functionality. So on this panel here on the right, you see this staining for example, the comparisons between young and old, and you can see that aged muscle is quite incapable or impaired in terms of its regenerative capacity. So the first part of my talk will focus on what is the effect of this aging niche on the alteration in transcriptome and epigenome, and by proxy on the regenerative function of muscle stem cells. This is a project that we started about 4 years ago, and it's led by my senior PhD student, Felicia Lazur. So one of the first questions we asked was, how does aging affect the population dynamic of muscle stem cells? We know from numerous studies published in the past that muscle stem cell is not just one cell type. There's a lot of heterogeneity in the muscle cell populations. And the questions we were having were, is it a subpopulation of muscle stem cells that gets lost, that leads to that reduction in the total number of stem cells? Or is it just a stochastic loss of everything basically going through the decline during aging? So the advent of the single cell technologies and single cell RNA-seq allows us now to actually look at the population dynamic of these cells throughout the aging process. So what we did, one of the first experiments was basically we're just looking at the C57 Black 6. These are mice that Jax was that Jax was aging them at the time for 18 months. I think they aged them now for 2 years, but they're quite expensive. So here we're just comparing the young and old mice. So we fact sort satellite cells, and actually we wanted to do satellite cells and as well as at least 2 non-myogenic niche cells, fibroadipogenic cells, which have been shown by Fabio Rossi's group and subsequently by other researchers to play an important role in regulating the proliferation and differentiation of muscle cells during regeneration, as well as macrophages, because macrophages are communicating quite extensively with satellite cells during regenerations through the secreted factors. So we isolated these 3 cell populations from the young and old mice, and the young is 6 weeks, old is 24 months. So that's throughout the course of my talk. This is the age, whenever I talk about aged or old is 24, and then young is 6 weeks old. And Felicia developed a very efficient FACS strategy where she could simultaneously isolate all this 3 cell population from the same mice. So essentially she was getting for alpha 7 positive cells and then lineage minus. She would then take the SCA1 plus cells, and then she would use the CD11B and then F480 for macrophages, and then put these cells and then subject these cells to the next genomic chromium 3' chromium platform. Then we would get the single cell data from there. She did a number of quality control tests just to make sure that she was isolating pure populations of satellite cells. For example, satellite cells express a transcription factor called PAX7, nicely stained for that, FAPS for PDGFRA, and then macrophages F480. So this is a count that shows the purity of satellite cells. She also did a post-sequencing QC where we look at the single cell data with satellite cells express satellite cell marker, which is PAX7. Then FAPS express FAPS marker, PDGFRA, and macrophages express macrophage marker. So we were certain that we were isolating pure population of these cells. So here, what I'm showing you is a UMAP plot. This is essentially a very reductionist kind of notion. This is almost like a PCA, but you all have seen this kind of plot. It's very similar to TESNI. So basically, we're taking spliced reads from individual cells after doing the initial processing, which I'm not going to go through in the interest of time. And essentially, we just wanted to see these populations in relations to each other. So you've got muscle stem cells here, you've got macrophages down here, you've got FAPS down here, which is separating as expected nicely. And even with this early kind of clustering with the UMAP, you can see that the young and old are actually separating out quite nicely, especially for the satellite cells. What we noticed, obviously, we've known that the satellite cells decrease in number during aging for many, many years. But what we've noticed that there is an increase in the number of FAPS in aging. And macrophages really don't change that much in terms of their numbers, but as I'll show you, well, I'm showing you right here, there is a slight shift in the phenotype, which is expected, and it's actually being documented in the field. So we could do all kinds of things with this expression data. We could, all the stuff that goes actually into the supplemental. I'll just briefly tell you what are some of the interesting things. So what we have found that the main component or the main contributor, at least between these 3 different cell types to the ECM, the extracellular matrix, are actually FAPS, despite the fact that FAPS actually increase in aging almost 3 times. Their contribution to the production of ECM matrix, so this is the matrix where genes decreases. And again, as I mentioned before, you can actually see a clear separation of the young and old satellite cells from this. Macrophages, they switch. You can see that as expected in young mice, they have the anti-inflammatory phenotype, and there is a switch from the pro-inflammatory phenotype in macrophages. So data was consistent with what we already know from the literature. So now we can take, now I'm just going to entirely focus on satellite cells. Now we're going to take our satellite cells and again use the spliced read to do subclustering to see what is the heterogeneity in the cell populations between young and old. And what you can clearly see here, we can distinguish at least 3 unique subpopulation of satellite cells in the young. So this is this yellow population here, we call it satellite cell 3, that's the smallest population, and then there's red and blue populations, which have distinct gene expression profiles between them, but I'm not going to list all the genes that are present there because there's something more important that I want to talk about. And what we found, which is the answer to the first question that we had, is the decline in satellite cell stochastic, or is it a clonal? And it clearly shows that it's a clonal loss of muscle stem cells during aging. For example, this yellow cluster is completely lost in the aging. There is a reduction in the red cluster, but as you can see here, if you look at the proportions, this cluster seems to be quite happy with the old niche. So it's not like, so there's a difference, and we can get into this in the discussion without getting too much into possibilities or speculations as to why certain populations persist in aging and then certain populations are actually lost. Whereas with FAPS, for example, we could actually clearly define two populations of fibroadenogenic cells, this yellow and blue populations. And interestingly, the FAPS, both populations actually increase in aging. So in contrast to the muscle stem cells, which follows this unique pattern of clonal loss, FAPS seems to be there is a general increase in the FAPS population. Not much of a difference in terms of cell number between macrophages, except for, as I mentioned, that subtle phenotypic switch between those two populations. So from this analysis thus far, we clearly see that the population dynamic of satellite cells is quite unique or different than the FAPS and macrophages as the mice ages. So clearly this yellow population is lost. But what is interesting for us is if we look at for example, this red population in young and then compare it to the red population in old. Essentially, these two populations are the same, or at least one is derived from the other. And then compare the inter-cluster gene expression differences. What you can see that there is a very clear and strong correlation between genes that change between these clusters, between these two clusters, between young and old. And interestingly, if we look at FAPS, and again, we could do this because we have more than one cluster. You can't compare one cluster to itself. So in FAPS too, we can do that. We can compare these clusters, blue to blue and yellow to yellow. And then we can clearly see that, again, this is a very nice correlations between the genes that are changing between these two clusters, even though they're actually expressing unique genes. So what is common between these clusters and between these two different cell types? Well, if they're within the same muscle, they're within the same mice, and they're undergoing the same process of aging. So our questions at this stage was, are these changes actually induced by the niche environment, or are they because, you know, if you're dealing with the effect of age on stem cells, almost everything has been associated with aging, from mitochondrial dysfunction, oxidative stress, DNA damage, you name it, the list goes on. But what was our interest at this point? We wanted to know if these alterations in gene expression are actually induced by the altered niche environment. And so to do that, we needed to develop some in vivo models. So these were some of the questions that we asked. Are the alterations in satellite cells transcriptome reversible? Can be. So if the niche is changing these alterations in the muscle stem cell transcriptome, well, if we modify the niche, or we do it through the cell transplantation, for example, can we reverse some of these genes? Are the alterations due to the post-transcriptional or transcriptional mechanisms? It could be at the level of RNA stability, processing, and transport, or it could be at the level of chromatin and epigenetic mechanisms. Are the alterations gene or locus specific or random? Is everything subjected to the, sorry for my this mouse is jumping. Next. Are the alterations gene locus specific or random? As in other words, you know, if there is a locus specific effect on an altered transcriptome or epigenome, if that is the case. And then the last question that we wanted to do, and through our model, so we wanted to design a model where we could actually quantify the direct effect of niche on the alterations of transcriptome and epigenome. So with this in mind, we designed this models here. So we needed to have, so we had to link three pieces of tools and technologies together. Obviously, one of them is FACS, which is described extensively in the literature, especially for the muscle stem cells. You can use GFP reporter, you can use cell surface marker. And then we needed to have an in vivo stem cell transplantation model where we would take cells, for example, from the old donor, put it in the young, and then take from the young and then put it in the young as a control. Then we could computationally model the effect of transplantation on the transcriptome, and then use that to specifically identify what is changed by the niche. And really what has been a game changer over the past few years has been the introduction of the SMART technology, which is the technology based on template switching, which is also used in the single cell RNA-seq. And tagmentation-based Nextera technology, where you use TN5 to actually generate libraries for both ATAC-seq and also RNA-seq from very minute amount of cell material. As you will see in my talk, we have generated RNA-seq library from as few as 30 cells. And this is important because engraftment is extremely inefficient. So what we do in our model, we have the old mice. This is old PEC7 GFP mice, young PEC7 GFP mice. We isolate satellite cells from these mice. We take 1,000 satellite cells freshly sorted at the time of isolations, and then we subject this to the smart RNA-seq analysis. And then we transplant 10,000 of these cells into the recipient mice, and then we let these cells stay there for 3 weeks to be exposed to different niche environment. We then re-isolate these cells, and then we do again bulk smart seq to see what has changed and what has remained the same. So I just want to, just so that everybody is on the same page, I'll spend 1 minute on explaining how the smart technology work and why is it so potent and so efficient for low cell numbers. So essentially, you don't need any RNA extraction protocols. You basically put your cells directly into the smart reaction buffer, and what happens there, it just blows up your cells, so the content of cytoplasm are out. So essentially what you do is use a poly-DT primer. This poly-DT primer is the same way you do the RT reaction. So the poly-DT primer combined with MMLV reverse transcriptase, and that's where the trick is. MLLV, when it goes to the end of the template, it adds few non-template nucleotides, which invariably is a bunch of Cs. So this is the trick. Then it eliminates the need to do any kind of ligations. You basically design your template switching oligo, this time with overhang GGG, which will anneal with CCC. And then within this, you can use overhangs, put Illumina adapter PCR primers, whatever you want. So now you have the priming site on both sides. So essentially everything that you do downstream is PCR driven, it's template driven. So the sensitivity of this technology is exactly the same as sensitivity of PCR. You can do, just showing you here how it can be done, but for ChIP-6 is essentially the same thing, except that when you do the chip DNA, you turn this DNA into single-stranded, use T-tailing, add a bunch of Ts, and then instead of going with oligo-DT, you go with poly-A primer, and then repeat this whole process again. So there are multiple versions of this technology that have been applied to the analysis of minute amount of material. So coming back to our model, here we have the old mice, the young mice, which we're transplanting into the old into young, young into the young. And essentially what we needed to do is to make sure that these cells are actually going into the stem cell niche, that these cells are actually coming from the donor. And then so with the GFP, we can trace them. With PEC7, we can make sure that there are stem cells. So what we do here, we irradiate this host mice to get rid of the endogenous stem cells to eliminate any kind of competitions from the endogenous cells. And this graph plot shows you here that we actually deplete this quite significantly, the endogenous cells. And then we inject this donor cells, and then we do a lot of cross sections to make sure that our donor cells are actually homing to the niche. They're in the correct niche location. They express PEC7, they express GFP, that's the donor cells. And then we can re-isolate them by effects. So typically we get about 100 to 200 cells after engraftment. And then so at the time when we did this experiments, we would call the genome center and say, I have 200 cells, I want to do an RNA-seq on that, and they would tell you, forget about that. So we actually spent a lot of time, and this was thanks to Felicia and then 2 other PhD students in the lab. They troubleshooted every step of the process to generate the sequencing ready libraries from this minute amount of cells in the lab itself. So that just it's just a flow that tells you what we needed to do. So cells are fact sorted, they go in the smart lysis buffer. Then we do smart PCR. We actually generate cDNA. We use a first step of ampere purification. So we select for a specific size here that corresponds to the average transcript. Then we put Illumina indexed adapters using the TN5 and Nextera technology. So now we have segmented this, and then we do PCR again in the last step of ampere purification or selection. This last step is critically important because you want to get rid of all the primer dimer. You don't want to sequence Illumina adapters. So that's this is just an agarose gel that Felicia has shown here where she wanted to get rid of anything that is below 200 base pairs, but not touch the 200 base pairs. And in fact, this 177 base pair is an artificial band that she introduced into this DNA ladder by PCR because she wanted to make sure that she's just going under 200 and not 200 base pairs, which is part of the sequence. So here are just some of the bioanalyzer results that I'm showing you here that shows you that there's really no difference in terms of the quality of DNA or tagmentations as you increase or decrease the PCR cycle numbers. She came up with this table here for optimization. So if we had between 30 to 50 cells, she would do 17 PCR cycles, 50 to 100, she would do 16, 100 to 150, 15, and then the list goes down. So these are some of the IGV tracks for the RNA-seq data showing that yes, we actually get full transcript RNA-seq profiles of these cells from 30 to 1,000 cells, and it's a very high depth. And you know, in terms of people ask me how many reads do you get, it depends on how many samples you multiplex, but typically we obtain at least between 30 to 50 million reads, which is more than sufficient to do the differential expression analysis. So now we have this tool. Now we can ask the questions. If we take cells from the old mice and then put it in the young niche, because we know during aging, there's, depending on where you draw the threshold, there are hundreds of genes that change. So we can now go say, well, how many of those genes are actually restorable to the normal level? And now we have a control because we have a young-to-young control just to adjust or to computationally model for the effect of transplantation and FACS abuse, Hamilton's syringe. So there's a lot of stress that is put on these cells, but we can, from young into the young niche, we can use that as a background control for the noise. So what we find here, these are the genes that are actually changing. So here, young T0, for example, these are a whole bunch of genes that are repressed under normal conditions in the young mice. We know that the old T0, these are the genes that are actually going up. So yellow, red means it's going up, blue means down. But what is interesting that these genes, most of these genes are actually restorable to the original states. It's independent of just the transplantation. Remember, we're modeling the noise out. So this is great. So we actually see a lot of genes that we can restore back to the original level that they were. And these genes include, these are just a few examples. I'm telling you, COL3A, collagen, SPERC, for example, COL4A2, and MMP2, whether genes are going up or down. There are some genes that that follow this pattern that you can actually restore them back to their original state. However, there are a lot of genes that are changed in aging, and then they're resistant to the niche factor. So, you know, you take the cells from the old mice, put it in the young, it doesn't matter. We leave it there for 21 days, that's 3 weeks. That's, in my opinion, it's sufficient enough for any changes that can take place to take place. But these genes, for example, there's a whole list of them. They go, they change in aging, and they're extremely resistant, or literally they do not change by even after 21 days exposure to the young niche. So genes like, for example, IGF-1, P2M, beta microglobulin, for example, this altered in aging is also called an aging factor. Cyclin D1, these genes cannot be changed back. And then, so this is just a UCC track again, showing the RNA-seq reads. So what we, so clearly from here, what we know is that exposure to the young niche environment or the different niche environment can restore and I'll give you the number, the exact numbers, how many genes, depending on the criteria, how many genes change in aging and how many of them are restorable and how many of them are not. But clearly what we have seen here, that there's a big difference, seems to be a difference, seems to be a difference. And so remember I said, you know, we wanted to know if all these alterations are just random or locus specific. So it seems like certain genes are responsive to the niche and other genes are not. Now, at this point, we wanted to know, well, all these alterations, whether they're induced by aging or whether they're induced by the niche, are they actually at the level of transcription or at the level of chromatin, or is it at the level of post-transcriptional modifications, for example, the RNA stability, because we're at the fork, we have to decide. If it's at the level of post-transcriptional modification and RNA stability, then we could go after RNA binding proteins and pathways. If it's at the level of chromatin, then we dig into the chromatin state, and then we'll look at epigenetic mechanisms that might be altered during aging and might be responsible for this process. So what we wanted to do was to first analyze the RNA for, or the altered transcriptome for the RNA stability. So the model that we have used here, this is from my collaborator, we applied this hot tub model. So in this model, the water that is coming inside the hot tub is the nascent RNA. This is the RNA that contains both intronic and exonic reads. The water inside the top is processed RNA. So this is steady state mRNA, and it contains exonic sequences, and the water that is exiting the top is degraded RNA. So degraded RNA is primarily driven from the mRNA sequences, meaning that it contains a lot of exonic sequences. So if nothing is changing, the ratio of exonic to intronic RNA for every gene will remain the same, will remain constant. If a gene changes in terms of its stability or degradation, then this ratio of exonic and intronic RNA will shift. And so we could use mathematical models to actually predict that. So the way it works, it's along this diagonal for every gene, every blue dot represents a gene. If there is no shift, you would expect that everything falls through this line here. If there is any major shift between the two conditions, the degree of this shift will represent the stability or the degree of destabilizing effect on that particular genes. And this is just a mathematical model that explains it. So when we apply this to ourselves, for example, to our aging model, when we look at the age effect on transcription, an age effect of mRNA abundance, we see again every gene or the data clearly falls on that diagonal. So there's really not much. I mean, you could find some genes that are changed during aging, but globally, when we look at it, it seems like changes in transcriptome during aging are not the result of RNA stability. So there must be something else. Now, when we take the cells from the old and we put it in the young, and then we see which genes have been reversed back. So the altered transcriptome by the niche, again, we see this very nice correlation in terms of transcription, but we don't see anything in terms of stability. So you just see this blob right here. So it seems like post-transcriptional modifications do not seem to be playing any significant contribution to the altered transcriptome between young and age, or in the reversal process that we expose to the niche. It must be at the level of chromatin and epigenetics. So another PhD student in my lab, Corinne Shahinian, so we deal with satellite cells, and we deal with satellite cells in, for example, in aging, disease models. And then so any kind of technology that there is genomite that could be applied on the genomite scale, we need to really scale it down to the nanoscale. Like for example, in the ATAC-seq, yes, there's ATAC-seq works great when you have millions of cells. But so I tasked Corinne to develop an ATAC-seq protocol based on only ATAC-seq to modify it so that we can actually get the results from 5,000 cells. And then she spent quite a few months on it, but she was able to optimize it quite well. So we performed ATAC-seq on satellite cells, the freshly sorted satellite cells from the young and old mice. And not surprisingly, since the RNA stability wasn't a factor, you can clearly see that there's a big difference between young and old. So they're separating out well, meaning that chromatin is altered during aging. I'm just showing you a couple of genes here, just as an example. For example, CMA3A, which is a secreted factor. Here, for example, in the young, you have the TSS transcription start site of the CMA3A quite open. In the age, it's actually closed down, and some other genes too, whereas the control or the housekeeping genes do not change. So if we separate those, just coming back to, remember I said there are differences between genes. There are some genes that are responding to the niche, and there are some genes that are not responding to the niche. So if you're comparing, my mouse is playing tricks with me. If you're comparing young and old or any two conditions, you will have genes that are going up, you will have genes that are going down. Now we're transplanting cells, and then we'll look at what genes are changing and what genes are not changing. So then you will have these categories. Genes that are down but are irreversible, so this red dots. Genes that are down but they're reversible by exposure to the niche. And this green dots, genes that are up irreversible and genes that are up reversible. So we can actually divide our genes from the young to age and engraft it into these four categories. And clearly what we see that down irreversible genes have a very low accessibility of chromatin. This is not a very visually impressive figure, but almost 70% of the red dots are actually below this zero line here. So now, coming back to the numbers that I promised to give you. So if we take a, draw a p-value threshold of, or adjusted p-value of 0.05 log-fold change of 1, we see between young and old, about 899 or 900 genes are actually changing. If you look at overall how many of those genes can actually be restored back by exposure to the young niche, pleasantly surprised that more than 50% can be restored. But this is where it becomes tricky and very interesting. Genes that are up in aging, almost 3/4 of them are actually restorable. So 73.4% of every gene that goes up in aging can be restored to its original level just by exposures to the young niche environment. However, if you look at genes that are down in aging, this is very different. Most of the genes that are actually going down in aging cannot be restored to their normal level by exposure to the niche. So simply means that, and remember, this is not because of the RNA stability and post-transcriptional mechanism. So simply means that the response of a gene to the niche environment must be dictated by the state of the chromatin. And you can see also this very clearly, the same thing in the scattered plot, where you can see this very skewed kind of distribution of those genes. So the color indicates the effect of engraftment. The more purple you have, the more you can restore them back to its original level. So clearly genes that are up in aging are more prone to be restored back to its original states compared to the genes that are down in aging. Similar results can be shown here in this volcano plot. So for example, this is our control. You would expect genes to be on either side of this line. So this is our age between young and old. And then you have this very strong skew here, suggesting that the response to the niche will depend on whether the gene is activated by aging or repressed by aging. If it's repressed by aging, it's very unlikely to be restored back. If it's activated by aging, it's very likely to be restored back to its original state. So this diagram here basically summarizes what I just told you. The degree to which gene expression response to the niche depends on really the chromatin states. Repressed chromatin or repressed genes are not responsive to the niche environment. So then we can look at more specifically, well, what is so different between active genes and repressed genes in terms of their response to the niche environment? Well, when you have active genes, you have the promoter open, you don't have any, sorry for my mouse, it's playing tricks on me, this Apple mouse. You have open TSS, you have engagement of RNA polymerase II with transcription factors. You essentially have no DNA methylations within that promoter if it contains CpG islands. And from here on, when I talk about DNA methylations, I'm talking about methylations within the CpG island. When your CpG island promoter is methylated, obviously you will have metal binding domain proteins binding to the metal groups, and then that will act as a platform for recruitment of histone deacetylases, this chromatin modifier, that will block the activity or the binding of transcription factors to that promoter, and essentially you'll get this in a very stable repressed state. There's also a different kind of repressions, which is DNA methylation independent. For example, here it's shown by PRC2. So we ask the question, well, if the repressed genes are not responding to the niche environment because they're methylated, then we should see a difference in the distribution of CpG islands. If you just look at the, if you take all of the genes that fall into those categories, then we should see a difference in the distribution of CpGs associated with the promoter, and that's exactly what we see. So here, if we just look at the up or down, just put them together based on irreversible or reversible. If a gene is irreversible, you have a higher frequency of association of that promoter with CpG island compared to the reversible. Now we can further look at this onto down-regulated and up-regulated genes. And in the up-regulated genes, which there is no difference between reversible and irreversible in terms of their association with CpG island promoters. However, in the down-regulated, remember, these are the one resistant to the niche environment, you see a very large difference between what is reversible and what is irreversible in terms of the frequency with CpG islands. Down here, this plot shows it even better. So these are the overlapping with the TSS versus within the 5 kb of the TSS. And again, you see a big difference between when it's reversible or irreversible in terms of having the association with CpG islands. So we think that the The fact that repressed chromatin is resistant to response to the niche is because these genes, A, they're associated with CPG islands very significantly, as I showed you some stats. And then B, once they're methylated, then pretty much they become resistant to the heterochromatizations by DNA methylation will likely be irreversible by the niche factor. So we have performed DNA methylations using two different strategies, using the MIDIP-SIC, which uses an anti-methylcytosine antibody, as well as we have done the whole genome bisulfite sequencing, but we still haven't received that data to really conclusively show that, that what we see in terms of correlation and association and increasing frequency is actually due to the methylation. So that data, unfortunately, hasn't become available. Things are going slow because of COVID issues. They're still sitting at the Genome Center. So from what I have told you from part one, so far what I can summarize is that aging leads to a reduction in the diversity of satellite cell pool. I think our single cell data clearly shows that reduction in the muscle stem cell pool is not stochastic. And age effect on muscle stem cell transcriptome are primarily through transcriptional mechanisms and epigenetics and not post-transcriptional mechanisms such as RNA stability. Age effect on muscle stem cells is largely reversible. It's more than 50% is reversible. However, the degree to which a gene responds to the niche is dictated by the chromatin state of its promoter. And we think that it's most likely culprit behind the genes that are resistant to response to the niche is because they're methylated at the level of DNA. So that brings me to the second part of my talk, which is going to be actually a shorter part, because this is a paper that recently published in McGill Reports. And then so in this paper, what we showed, there was a signaling interactions between skeletal muscle fiber and the satellite cells. So in this context, muscle fiber was acting as ligand source and the satellite cells was acting as receptor. And really this project, I got excited about this project because I do a lot of ChIP-seq and RNA-seq. Sometimes I just want to see, how does it look like? And then so I wanted to compare all four myogenic factors. There are only four myogenic factors, MIF5, myodemia, myogenin, and MIF6. I just wanted to know where they bind, how do they differ, within their own context, like MIF6, for example, is expressed in a fully differentiated state, whereas MIF5 is expressed in proliferating myoblasts. So I would do the ChIP-6 and, of these myogenic factors within the context that they're actually active. And then so, we knew that MIF6 is, perhaps because it's expressed in a fully differentiated states, it's probably the only function of MIF6 or the primary function of MIF6 is to maintain the differentiated state. So I was looking for MIF6 binding to structural genes that are required for the myofiber and whatnot. And then sure enough, yes, MIF6 binds them. But what came to surprise me was the presence of MIF6 binding sites within the genes that the fiber didn't seem to have any use for it. For example, these are secreted factors. It's epidermal growth factor, OSM, LIF, and VEGF-A, and a lot of secreted factors that didn't seem to be used by fiber in the first place. And then so, and these are considered myokines because they're produced by the skeletal muscle, right? So I asked one of my students, I said, why don't you compile the entire list of myokines based on literature and then see where this, how MIF6 binding sites around those myokines are. And this is the list that she was able to compile. And then you can see that the binding of MIF6, so the redder the color, the higher the number of peaks around the regulatory regions of those genes. And you can clearly see that this list seems to be containing a lot of MIF6 activity around the regulatory regions of those genes. And on this panel here, we're just showing gene expression analysis during the time point of myoblast differentiations in 5 days. And again, it's a big cluster of those myokines are actually activated at late phase of myogenic differentiations when these multinucleated tubes are formed. And those are the ones that correspond to a lot of MIF6 binding sites. So I want to make this story short because it's published, like I said, but what we found, so here, for example, this is just a heat map that shows you the expression of those myogenic factors between satellite cells and single myofibers. And when I say single myofiber, this is actually one EDL muscle fiber. We do the RNA-seq, whole transcriptome RNA-seq on single myofiber. We published this protocol in bioprotocol, detailed protocol of this, how to do RNA sequencing on a single EDL myofiber. And so we get rid of all the satellite cells by simply subjecting this fiber to trypsin digest, or treating it with trypsin to get rid of satellite cells, wash them up. And then what you get is entirely from the fiber, not the other non-myogenic cells. So here we see again, MIF6 is only expressed in the myofiber, not in satellite cells. But what we saw a pattern in a lot of those myokines was something like this very simple plot, for example, here. So here you've got satellite cells. This is just the gene expression. You've got satellite cells, myoblasts. These are proliferating myoblasts. These are early differentiating myocytes. And then myotubes. So for example, if you look at EGF, the epidermal growth factor, it actually comes up. It's not expressed in satellite cells, but as you differentiate these satellite cells into this multinucleated structure, myotubes, then you see an upregulation of EGF. That upregulation quite nicely corresponds with, obviously, upregulation of MIF6. At this point, I'm not saying that it's a target of MIF6, obviously it is, we have published it. But if you look at the receptor for EGF, for example, so EGF is just one ligand for the EGFR receptor. The receptor is expressed on satellite cells. And then as these cells go through differentiation, the receptor itself is not expressed. Then they switch to producing the ligand. So clearly this was suggesting to us that the fiber or the myotubes and satellite cells, they seem to be communicating, or at least there was a suggestion that they might be communicating through this receptor ligand interactions. And so this is just RNA-seq reads of EGF and EGFR between satellite cells and then myofibers, and again, showing the same pattern. So we wanted to see if there's any relevance of MIF6 regulating this myokines in the muscle, obviously in vivo, and what the effect would be on muscle fiber, on the overall body, the physiology of the mice, and on the satellite cells. So we got this MIF6 knockout mice from Christoph Lepper at Ohio State University. Yes, he's in Ohio. He used to be in Baltimore at the time. And so what we did, one of the first experiments we did was to isolate whole muscle RNA from injured and uninjured mice, and then look at the expression of those myokines. Keep in mind that a lot of other non-myogenic cell types might be contributing to the production of those myokines, but we just wanted to see if in the absence of MIF6, we actually do see a reduction in those myokines, that would suggest that MIF6 might be transcriptionally regulating those myokines. And that's what we see, not for all myokines, but for some key myokines, and I'll focus on EGF. But we have other ones in the paper as well, that when you get rid of MIF6 in vivo, you actually get rid of the expression of EGF quite significantly. So then we said, well, is muscle or the myofiber the main contributor of the epidermal growth factor in the mice? And then so clearly here you have serum from the mice. And in the MIF6 knockout, we have a significant reduction in the EGF level. And this is very important because there are 200 different, in the human body at least, there are 200 different cell types that could be potentially contributing to, many of them could be contributing to the expression of these secreted factors and their presence in the bloodstream. However, when we, in the MIF6 knockout, clearly in the serum, we have a very significant decrease in the EGF. This suggests that transcriptional regulation of EGF by MIF6 in the muscle fiber is the main source of the EGF in the blood. You could do this in vitro by differentiating myotubes from the wild type and MIF6 KO knockout, and then collect secretome, and basically it shows the same thing. So then obviously we can apply our single myofiber RNA-seq, and you can clearly see the muscle fiber expresses EGF. This is our MIF6, our favorite gene at the time still. This is our negative control OCT4, and then CKM, muscle creating kinase, clearly showing that you can get whole transcriptome RNA-seq from single fiber, and then look at whether or not the muscle fiber itself, in the absence of other non-myogenic cells, is producing the factor of your interest. That's just a cartoon schematic of how we applied this SMART-seq technology to develop to infer the RNA-seq from single fiber. So then we said, well, if muscle fiber is producing EGF and the satellite cells have EGFR, then there must be an impairment in the EGFR signaling in the satellite cells associated with MIP6 knockout. So we look at, the EGFR phosphorylation at tyrosine 1068, and then clearly in the absence of MIP6, we have a reduction in the number of EGFR plus, PAC7 plus cells. Interestingly, when we add exogenous, when we add EGF, we can rescue this, suggesting that this is actually a ligand-mediated signaling. This is not a sub-intrinsic defect in the satellite cells itself. So what happens to the muscles of this mice in terms of the myofiber morphology, the shape and size? We have analyzed them at P7, P21, 3 months, and 10 months. We really don't see that much of a difference. There might be a subtle difference between them, but not a significant one. However, what we found was, and it was puzzling, is that by 3 months of age, so even though you start with the same stem cell pool, so this is not a because this is germline mutation, right? This is not because of the defective establishment of stem cell pool. You clearly start with the same stem cell pool at P7, but then gradually you have the number of stem cells reduced at 3 months and then more so at 10 months. So the stem cell population is decreasing, and so what happens to them? They're either going through senescence, which we showed that this is SA beta gal staining. This is our positive control with hydrogen peroxide, and the comparisons between wild type and knockout, we don't see any difference between them. So these cells are not going through senescence. Then we look at apoptosis, and then again, this is our positive control with strasporin. And again, we don't see any difference between wild type and MIP6 knockout. So these cells are not lost to apoptosis, they're not lost to senescence, they're actually lost to differentiation. And so the way we found out, we isolated satellite cells from the MIP6KO1 wild type, again, using our SMART-seq technology. What we did was to look at pathways that are highly differentially regulated between the wild type and knockout, and the pathway that popped up was P38 MAP kinase pathway, which obviously induces MyoD, or activation of MyoD and induction of myogenic differentiation. These are myoblasts in vitro forced to go through differentiation when we add EGF or we add LIF. We actually block P38 MAP kinase activity, and then we block the induction of MyoD. So it seems like the production of this myokine in the muscle fiber is preventing the activation of these cells by P38 MAP kinase and induction of MyoD. Therefore, this communication between muscle fiber and satellite cells is critical to maintain them in a quiescent state. So this was in response to one of the reviewers who was adamant that these cells have sub-intrinsic defect. They don't. They actually go they have perfect differentiation, proliferation, and everything, is in par with with wild type cells. So essentially, what we wanted to do was to say, because we were saying that the niche is defective, it's not the cell. So essentially what we have here, we have this luciferase expressing mice, we isolate satellite cells, and then we have MIP6KO1 wild type, we irradiate this mice, we inject the same number of cells, luciferase cells of the same mice, into these mice, and then we monitor the luciferase activity by bioluminescence imaging for 21 days. And what we can see is that the MIP6KO muscle is incapable of maintaining muscle stem cells, or engrafted muscle stem cells, meaning that it's not the cell that is defective, it's the muscle that is defective. We can actually show this in vitro, exactly the same thing in vitro by culturing these cells with the various reporter cells. So essentially what we have shown here is that the signals coming from the muscle fibers, which are under direct transcriptional control of MIP6, primarily EGF, are blocking P38 MAP kinase activity, and that blocks myogenic differentiation. In the absence of this, you will have premature activations of muscle stem cells and reduction in the stem cell pool. And then so this was clearly establishing that the muscle fiber is not just a physical entity that the cell is sitting on, the satellite cells. It's actually a very critical component of the signaling component of the muscles. So this brings me back to, sorry, I'm a bit, I guess, over time, but this brings me to the most important slides. This entire experiments that I described to you were actually done by these three great PhD students. They're still currently in my lab. Felicia Lazur, she led the aging project. Darren Blackburn with the MIF6 in collaborations with Felicia. And the entire chromatin work that I described to you for all the project was done by Corinne Shahinian. My collaborator, Hamid Najaf Al-Adi, is a computational biologist, and Rick Ferroni did a lot of analysis for the single-cell RNA-seq data. Yanis Rogosas at McGill University, who was just basically charging me for the reagents, great collaborator at McGill for single-cell RNA-seq. The C3G group at McGill Genome Center, again, for a lot of computational analysis of the attack-seq data. Christoph Lepper gave us the MIF6 knockout mice and did a few initial experiments there. So Colin Crest, we used his expertise for the cell transplantations. And Arizu Jahani, my neighbor, she has been collaborating with me on pretty much on all of the projects that or both of the projects that I described to you. And then my long-term collaborator, other computational biologists. So I'm surrounded by a lot of good computational biologists. Ted Perkin is one of them for the MIF6 project. And a lot of reagents that I used, especially in the MIF6 project, came from my former mentor, like Dr. Michael Rudnicki in Ottawa Hospital. And these are just the funding agencies that I need to thank. And thank you very much for your attention. I'll be happy to take some questions now.
Excellent. Vahab, thank you so much. That's beautiful slides, and what a great presentation. And you're obviously in a great environment there, and the technology is very, very impressive. Thank you very much.
Thank you.
So I'd like to invite some questions through the question and answer panel. I have a few that have come up, and I'll read them out.
Sure.
If you have some more, please add them to the list. I'll just ask one myself from
Sure.
a very naive perspective. One of the most interesting things you spoke about was the reversibility of the aging gene expression in those cells. And I mean, a lot of people would be very interested in hearing about that. And I wondered if, you know, some of the technology that you have applied and are expert at could be applied to a situation like exercise, for example. And I mean, it'd be fascinating to see if a physiological condition like exercise could reverse some of that gene expression that, you know, is part of the aging process. What do you think?
Absolutely, yes. No, so this, you're right. I mean, these tools and technologies that we have, I mean, we took I'm not taking any credit for SmartSeq or for the Nextera, but as far as I know, when we started, and I think even now, my lab is the only lab, I think still, who has taken these rare cells from a different host and put it in a new environment and then re-isolated and actually profiled not just transcriptome by the epigenome of this cells as well. So one of the things that we wanted to do was just to develop a model not just for the muscle, not just for the aging, but for example under exercise or under muscular dystrophy conditions, for example, or for the cachexia, for example, in sarcopenia, any kind of niche that you can imagine. This technology, these tools could be applied to. Yes. And exercise is one thing that I really, because especially with regard to the myokines, now we have a genetic model that if you get rid of it, you basically abolish or at least significantly reduce the expression of a lot of myokines, not EGF, you know, VEGF-A, for example, is one of them. And then so can we use that in an exercise model? And then what is the effect, for example, of this lack of myokines in this model on the population diversity of not just stem cells, other cell types, for example, yeah. Right? Yeah. And yeah, so I totally agree with you. That could be applied to multiple models.
Yeah. I bet you that would be a great point for collaboration if you were interested. Yeah. So let me get to the question and answer list here. Fabio Rossi asks, how long are the transplanted cells in the recipient animal before being re-purified for sequencing?
So they're there for 3 weeks, 21 days. So that's the day we inject, and then we wait for 21 days. We re-isolate them. And when we isolate those cells, as they come out of the fax, they actually go into the smart lysis buffer. So we don't allow them to sit around on ice or anything. So every single cell that comes out of the fax from the first cell to the last cell, goes straight into the smart lysis buffer, and that's destroyed. And then we do the cDNA synthesis there using the template switching platform.
Very good. Mehdi Seno asks, sorry if I missed this. I'm wondering if the mice used as hosts for the new cells were immune competent, and if so, how the immune response against them was controlled.
Yes, exactly. So they were not. These are NCG mice. These are immunocompromised. I was expecting at least, I'm expecting a question. So, if there is also immune component to the stem cell niche, right? If these cells don't have B cells and T cells, the mice don't have B cells and T cells, well, how would that affect the engrafted cells into, because we're putting the donor cells into a host that is immunocompromised, right? But unfortunately, with the wild type, it's very difficult to get that kind of a engraftment efficiency that you get with, for example, like NCG mice. We could control some of that by, I guess, by low throughput